All right, good morning. We are heading into our second to last week of the quarter, and this week our topic is on inheritance and genetic disorders. The question for the week is: how can two healthy parents have a child with a genetic disorder? Focusing today on how a mutation in DNA changes the structure and function of a protein and therefore the cell. So, our vocab words are all over here. We have seen some of them before, but we're going to go into more depth about them. So pushing forward, just a reminder, second to last week of the quarter, focus of this week is how can two healthy parents have a child with a genetic disorder? So on Monday, we're going to talk about outcomes of genetic mutations. Tuesday, genetic variation, reproduction, and inheritance. Wednesday, there's no school because it's Veterans Day. And then Thursday is inheritance patterns, so dominant versus recessive. And then Friday is genetic disorders and wrapping up your revise and EQ check. So for today, let's go back to the essential question. Let's write down some of our initial ideas. So, what is the cause of a genetic disorder? How can somebody get it? What makes genetic disorders different from diseases like cancer? What are the two types of reproduction and how do they vary? And how do living things pass on traits? How much DNA and chromosomes are passed down from a parent to a child? So remember, just write down whatever you know about these questions in your EQ checker. After you do your initial ideas, let's talk about how do changes in DNA lead to diseases. So a disease is any condition when the body is not functioning properly. Last week, we talked about how do healthy cells become cancerous. So what happens is that changes in the DNA occurs after the development, which is where cancer is developed. This week, focusing on how can two healthy parents have a child with a genetic disorder. What we are discussing is changes in DNA that are inherited, so genetic disorders. So that means during the process of the development of everything. So last week was after development of cells and everything. This week is during the development of different cells. The next week is our last week, and we're going to focus on health, disease, and treatment, and as well as your project. So, what is the root of a genetic disorder? Genetic mutations. So last week towards the end of the week, we started to discuss mutations about how if the DNA sequence is broken, that is when the mutations happen. So they are also the origin of all genetic variation. So what makes us all look different and are different. So without mutations, all life would be the same. So not all mutations are bad mutations. So a mutation is a change in the DNA sequence of a gene that can also cause or change the protein it codes for. So normal gene, mutated gene, we saw that last week that creates an abnormal or no protein whatsoever. So any daughter cells coming from a mutated cell will also have the mutation. So what causes a mutation is there is a mistake made during the DNA replication step of the cell cycle. Environmental factors such as radiation can also alter the structure of DNA, causing a mutation. And then there's a video linked right here on how radiation, like UV lights, gamma rays, X rays, can cause cancer mutation. And then if the mutation happens early in development or in sex cells, it can be passed on to the offspring. Okay. So, how do mutations lead to diseases? Remember, this is the process of DNA. We have, or of cells and proteins being created. We have DNA that gets transcripted, translated by RNA, and then they form amino acids. And as they fold, they then become proteins. So, if there's a mistake in the DNA, there will be a mistake in the RNA, which then means that there will be a mistake in the protein. So, it is what is it called? A ripple effect. So as it affects one thing, it affects everything. So here is a video. I'll let you watch this on your own about DNA and protein assembly. But protein synthesis is the formation of proteins by using information contained on genes, DNA, and carried by messenger RNA. Transcription is when copies of the instructions from DNA get transferred to messenger RNA. Translation puts together the chains of amino acids by reading the messenger RNA code. So remember, DNA is the original blueprint, messenger RNA deciphers it, and then turns it into the code needed to create the proteins. So transcription is part one. So making messenger RNA copy of a gene code for a protein. It's inside of the nucleus. Remember, DNA cannot leave the nucleus. What's involved is a gene, so a portion of the DNA that codes for the protein. So we just take one portion, or it just takes, not us, one protein at one section of the DNA code at a time, and it deciphers it. And then that's what codes for the different things. And then messenger RNA is a single-stranded copy of a gene. And then there's what happens then is that DNA gets transcribed. So DNA unzips at the location of the gene. So it comes apart because remember, DNA is a double helix. And then base pairs are matched to the template of DNA, forming a messenger RNA copy. So if you look right here, here is, let's say we took this section, and this is the first section of DNA. It matches up with its base pairs. Remember, we talked about those a little bit. So once it gets the base pairs all matched up, that is the code that gets sent outside of the nucleus. So messenger RNA breaks off from DNA and then leaves the nucleus, which happens right there. Okay. And then on your EQ tracker for refined task number 1, you are going to complete the protein synthesis for the mini gene below. So here is the DNA code that got unzipped. You are going to decipher it by using this right here. Remember, the key thing to remember is that thymine, which is the T, is only in DNA. So if you need to use the T or match up with the A, it will be U in messenger RNA. Very, very important. Okay, part 2 of this, so after transcription, we have translation. So the purpose of this is to make a protein from the messenger RNA copy of the gene. It happens in the cytoplasm at the ribosome, and what's involved is structure that performs translation. Messenger RNA codon groups, are groups of 3 bases that code for the amino acids. And then translation RNA or transfer RNA are 3 RNA base pairs with matching messenger RNA codons. So we have tRNA and mRNA, transfer and messenger, which carries amino acids and drops them off to make proteins. So remember, messenger RNA decodes from DNA, transfer RNA turns it into amino acids to create the protein. And the amino acids are the building blocks of the proteins. So then messenger RNA attaches itself to the ribosomes. So you can see that happening down here. Transfer RNA carries the amino acid pairs with complementary messenger RNA codons. So the amino acids are dropped off and bonded together, forming a chain. So you can see that all happening here. So it gets dropped off, gets transferred, becomes amino acids, and they drop off and become proteins. And then the stop codons ends the process of the amino acid chains and breaks off and folds into the protein. So there are different stop codons that at the end of that section, it breaks off so that that area gets coded for that specific protein, essentially. So remember, codons are 3 bases sequence on the messenger that RNA that codes for an amino acid. So based off of these 2 different diagrams, right? Based off of what it is. So let's say, so these are all 4 of the bases. Say my first code is UGA. So U is my first letter. I would go to the U, I would go to the G, and then I would go to the A. So this right here, there's nothing here. So that means that's a stop codon, right? If it was, let's say, UCA, then my protein that I'm coding for is SERT. So I would take that and I would write that down. And you can also use these as well. So If my first one is U, my second letter is C, and then my third letter is A, I would just go down here. And again, my amino acid would be SIR. So stop codons are 3-base sequence that ends protein synthesis. So here are the different codes for the stop codons. And then I will attach, this is Miss Buckles, but I'm going to put in mine as well. And then step 2 is translation, remember, is using the amino acid code table to find the amino acids that is coded by the 3-base messenger RNA codons. So this is part of your refined task. You're going to write down 3-letter abbreviations in the row that says amino acids. So under each messenger RNA codon, if you hit this, remember that stop, then the stop translating the sequence. That sequence is all done. Okay. What are ways that DNA can be mutated? There's substitution. So if this is the original sequence, remember each of these base pairs can only match up to another specific base pair. So if you look right here, here's the original sequence. This is the mutated sequence. So substitution means that instead of the right base pair there's something else that has slitted, has, taken or replaced it. Insertion is if this is the original code, there is a random code that gets placed in, and deletion means we're missing part of our code. So replaced, inserted in, missing code. So extra, missing. And then what you're going to be doing for today is you'll be doing the transcription and translating for a gene that has a mutation, seeing how it affects the protein. So finish the process of the transcription and translation for a second mutated sequence on refined task number 2. So number 1 was doing it for a healthy one. Number 2 was a mutated one. Describe the change in the mutated gene by saying what happened with the small mutation. So is it insertion, is it deletion, or is it substitution? And if you need extra help, there is a video that goes a little bit more in depth and gives more context on mutations. And as always, if you need help with anything, just let me know.